performance, how does the input relate to the output? Dynamic performance, how does this system behave over time? How quickly does it respond to the signal? Reliability, is this a genetically stable system or will it be lost over time? And finally, conditions. Again, what organism is it made for, and is it temperature dependent, and so on. So when I talk about static performance, I mean like a transfer function. If you have a system that reacts to an input signal and gives a certain output, how do these relate to each other? Because it's usually not linear in an infinite interval. Usually it looks something like this. That first, so this is the input signal, in this case a small molecule that activates this system, and this is the output. That first, nothing happens, and then you're in the dynamic range where the change happens. And then above this signal, nothing more happens. And this is things you want to know if you want to put this in a bigger in the context of other parts. What is the dynamic range of this part? Also, when you put things together, you want to know how quickly does each step react. If you have many parts in a row, you want to know how long time does it take before they actually do something. So that's the dynamic performance. So if you have, this is the output again, and this is time this time. So when you initially don't have any input, and then you activate, then you increase the input to a certain level, and then you just measure over time how long time does it take before we get a signal out of this. That was lots of theory. Before I end this, I will just mention some real-world applications of this. So it's a quite new field. So far there hasn't been that many real-world applications, but one of the most promising, and I will say a bit about, and that is an anti-malarial drug called artemisinin. As you know, malaria is a huge problem in the world, especially in Africa and Southeast Asia. And there's an anti-malarial medication. They're often quite expensive, and there are a shortage of them. So to alleviate this problem, some synthetic biologists tried to see if they could, instead of producing this medicine in the herb that it's usually from, so it's called sweet wormwood, they wanted to see, can we move these genes over to yeast and metabolically engineer the yeast so it will produce this drug? Because then you would not be dependent on seasons, you would not be dependent on the certain growth conditions these plants require, but you could grow them in tanks anywhere, right? So that's exactly what they did. So they managed to engineer yeast to make this drug, and now they're actually building a plant in Italy where they will start large-scale production of this drug this year. And they estimate that already next year they will be producing more than 50 tons of this drug annually through this synthetic pathway. So it works. I would also like to say a few words of my own research. So I'm working with antibiotic resistance, but the Synbio project I have is an attempt to reprogram bacterial immune systems to recognize, instead of fighting, so bacteria have immune systems similar to animals and plants, that they have to be able to fight infections of viruses. But what I'm trying to do is to see if I can reprogram these systems to, instead of identifying viruses as hostile, they should identify resistance genes as something hostile. And if this would work, so that's what a CRISPR system is, that's these immune systems. If this would work, might be possible to make bacteria get rid of their own resistance through their own immune systems. So I'm building now this synthetic CRISPR system that is targeting multi-drug resistance plasmid that was found at the Uppsala University Hospital a few years ago that caused an outbreak there. And so far it's looked promising, but we'll see how it will work out. So what is the future of synthetic biology? Well, the sequencing of organisms from all over the world is continuing. More and more genetic data is being generated through these projects and end up in our databases. So this means that we get more and more potential new parts from all kinds of life. And we also have making progress in protein engineering, and that will make it possible to engineer these natural parts, and it maybe even create completely new proteins that will make new functions that can do things that have not been able to do in nature, or improve existing function. And together, these things can have a wide range of applications. It can lead to new biomaterials, new drugs, energy energy production and storage, like biofuels. It could have medical applications, like the one I just mentioned, biosensors, and what was mentioned before, that's what we'll hear more about later. Eventually, we might be able to know so much about biology and the parts that make up biology that we could construct a minimal cell from well-known parts and build it from the bottom up. And that would be the first time man has created completely synthetic life. Finally, I would like to end with a quote from an American physicist that I think suits synthetic biology very well, Richard Feynman, that said, "What I cannot create, I do not understand." Thank you all for listening.
Okay, thank you very much. It was a very interesting talk. So now that you have all learned a little more about what biobrick is, what a genetic part is, and how you can use them to create new systems, we will, after the break here, the IGN teams will present what they have actually been able to do with the help of biology.